Thank you very much. It's truly a pleasure and a privilege to be here today and thereby have a chance to share with you a glimpse into what might be the future of organ transplantation. These are my disclosures. The idea of transplanting organs or other body parts seems to have always been a dream and one of the wild fantasies of mankind. And it eventually has turned into one of the greatest success stories in medicine of the past century. Owing to constant and new developments in surgical techniques and more potent and more sophisticated type of immunosuppressive agents have allowed us to perform more and more complex type of transplants over the past 60 years. These advances have also enabled us to continuously improve graft survival rates to highly acceptable levels across the board for most type of transplants. According to a current report, there have been about 2 million years of life saved in 25 years of organ transplantation in the US alone. This remarkable track record of achievement and the potential to do even more in the future actually are the reasons why organ transplantation has been listed among the top 10 medical advances that have and in many ways will continue to change the world today. However, those facts are sometimes easily forgotten as transplantation has become so routine and standard of care. But as we are looking towards the future of transplantation, I think it is critical to always bear in mind the pioneering successes and achievements that have made transplantation possible. Especially as there are still way too many patients such as Ellen, who is dying of organ failure, and she may or may not receive an organ in time. Unfortunately, we are still facing a still increasing disparity between the prevailing rate of organ donors and the patients in need for a transplant. And this will be a problem that will be with us for the foreseeable future. Also, access to transplantation is still not equally distributed, and it remains a privilege for those living in wealthy or technologically highly developed nations. For SIMU, for example, there might be not even attempts made to find a matching organ due to logistical constraints. According to statistics from the World Health Organization, organ transplants are currently meeting less than 10% of the global need, which is another major issue for the future of our field. And then there are patients such as Sonia, who has cancer that is curable, but her ovaries will be damaged by the required chemotherapy. She will most likely never have children and will suffer health issues related to hormone imbalances. Or patients such as Mark, who lost both of his arms while serving his country, and he would like nothing more than to receive successful arm transplants. And it is patients like them who are looking anxious towards the future of transplantation and what it might hold in store for them, and how innovations and technologies might translate into new treatment options and advancements in transplantation. Today, more than ever before, we're seeing scientific breakthroughs and we are living in times of scientific discovery. By unlocking the genetic code and developing technologies for gene editing, we will be able to treat and prevent disease that could not be imagined and will touch the most fundamental aspects of human life in the future. Similarly, advances in stem cell research and therapy or advances in tissue engineering, they hold tremendous potential to address the issue of organ shortage and actually lead to paradigm-shifting change in transplantation. In addition, robotics and artificial intelligence is getting more and more incorporated into medical practice. Implantable sensor or micro-robots are a reality and they are currently used for medical diagnostics and as innovative ways to apply therapy and therapeutics. And with the development of various different organs on a chip, we have actually set out to mimic whole organ physiology and eventually recreating life. So with all these technical advances and possibilities at our disposal, it somehow feels as if we are standing there again like Lindbergh at Roosevelt Field, ready to depart to new horizons in transplantation. But to what new lands might this journey actually take us? Well, it is always a losing position to be and to be the one asked to make predictions, in particular about the future. But one area that I think where we will see tremendous advancements is with regards to novel procedures and with new types of organ transplants. One such area is reconstructive transplantation that has been shown over the past couple of years to be able to replace both form and function in patients with devastating tissue loss, and thereby has become the latest chapter in this amazing success story of transplantation, with now hand, arm, upper extremity, and face transplants performed highly successfully. And as those images from one of our most recent hand transplant patients at Johns Hopkins show you, VCA is not only able to replace two arms, a face, or other tissue defects, but it provides these patients with a new outlook on life. And I think it's fair to go to even so far to state that VCA is currently the only restorative option available that is able to replace like with like. And by being able now to replace like with like, we can actually restore patients after amputation or devastating tissue loss back to normalcy. And in the case of face transplantation, VCA has allowed us to reintegrate these patients into a normal and productive social life. I would like to share with you this video from the Mayo Group, who will impressively show you what we can expect with regards to functional outcomes in VCA in the future.
Thank you so much, Gerald, for wanting to share our face transplant case from Mayo Clinic. This has been a culmination of many years of preparation. We want to share with you a small segment of a video that we prepared with Andy's story. We want to also thank you for your great contributions to the field of VCA and look forward to seeing you soon. The entire lower face, including the nose, the jawbones, the zygoma bodies, the skin all the way in front of the ear, down to the chin, and the teeth. They would have cutting guides for us that we would clip on the bones. They would give us the exact location of the cut, the exact angle of the cut. So when we took the donor's face and put it on the recipient, it would fit perfectly. Just here, you take the mirror.
After 3 weeks of healing, Andy was given his first glimpse in a mirror.
Wow.
Still recovering and not yet able to speak clearly, Andy scribbles his first impressions on a notepad.
Far exceeded my expectations. You don't know how happy that makes us feel.
Without the donor and the donor family, none of this is possible. I can't say thanks enough to those people for what they did, what they gave me. And now with this transplant, I just feel more comfortable and more confident in doing these things.
That's easy to understand when one considers the face that Andy now finds looking back at him in the mirror.
I can go out and I feel 100% normal.
Andy Sandness is able to look forward to his future.
However, despite those remarkable successes with regards to functional outcomes, the requirement for long-term multi-drug high-dose immunosuppression still remains a major limiting factor for more widespread application of VCA, which is certainly a life-changing type of procedure, but not a life-saving transplant. Based on these premises, our group recently developed the first protocol to minimize immunosuppression after VCA by using donor bone marrow cell infusions for immune modulation following CAMPF induction, and thereby we were able for the very first time to maintain VCA patients on a single drug immunosuppressive regimens at very low levels. Inspired by those achievements, actually currently more and more minimization protocols, including CNI sparing, steroid minimization, steroid avoidance, the use of biologic agents or other cell therapy protocols are actually introduced to VCA that will allow us to embark on even further frontiers in this field. So we have come quite a way to be able to minimize immunosuppression after VCA, but the ultimate goal would be to avoid all immunosuppression and to induce immunological tolerance for these patients. And some of those VCA actually, they include a far amount of vascularized donor-derived bone marrow and the donor bone marrow niche, which makes them unique immunologically and might favor tolerance-inducing approaches using chimerism-based protocols. With this hypothesis, we have recently developed a preclinical large animal model that combines donor bone marrow cell infusion with co-stimulatory blockade, and we were able to show for the very first time that we are able to induce immune tolerance and indefinite immunosuppression-free graft survival across a complete MHC mismatch in a preclinical large animal model. So with these advances, we think that eventually immune tolerance in VCA might become a possibility in the not too distant future. Similarly, although elusive for many years, immune tolerance in solid organ transplantation is a reality, with now successful trials ongoing at MGH, Stanford, and at Northwestern. And at Harvard and Johns Hopkins, currently efforts are underway to develop tolerance protocol in the setting of disease donation or in a delayed fashion. So with this prospect of actually tolerance become more widely available, we will be able to further expand indications safely in VCA, such towards pediatric transplants, as recently demonstrated by Dr. Levin and the Penn team, or even considering the transplantation of functional subunits such as eyelids, nose, ears, or lips, for which there is currently no viable reconstructive option available. Other areas that have seen tremendous expansion over the last couple of years are urogenital transplants, such as uterus transplants and most recently penile transplant. And the successes in uterus transplantation have been stunning, with now 8 live births after uterus transplantation in the last couple of years. So cell therapy will not only significantly shape the future of reconstructive transplantation, but it will also lay the foundation to develop several innovative therapeutic concepts that are currently underway in solid organ transplantation. One such prime example is the ONE study, which is a EU-funded, unified approach to evaluating cellular immunotherapy in solid organ transplantation. The ONE study team consists of various different centers from Europe and the US, working with a centralized laboratory to currently evaluate the safety and efficacy of various different cell products, with the main goal to induce monotherapy in combination with cell therapy. And although the results will be reported early next year, so far we can say that there is no safety concerns, that the tacrolimus monotherapy has been successful, and there is first evidence of clear benefits from reducing immunosuppression. And of course, we will see quite interesting results from the immune monitoring trials. Another exciting prospect for the future of cell therapy is actually the use of chimeric antigen receptors, or CARs, to generate T regulatory cells. Dr. Levin's group recently has shown the feasibility to redirect T regulatory cells towards a transplant-relevant antigen using CARs, and their group has also provided first in vivo proof of concept that such CAR Tregs can be used therapeutically. Ultimately, however, when we talk about the future of transplantation and bringing our field to new frontiers, then time will probably be our most precious resource. And such time to expand the viability of organs can currently be bought with innovative new strategies developed for organ preservation. There has been an entire revolution going on over the last couple of years with regards to perfusion technologies that have resulted ultimately in a whole new world of perfusion devices out there, including devices for lung, heart, kidney, and liver. And the impact of those technologies have been really impressively shown by the Toronto group, for example, where they were able to repair the organ from an extended donor criteria and then successfully transplant them, and ultimately significantly increase the number of transplants performed at their center. And similarly, normothermic liver perfusion from the Oxford group has shown impressive data in their first in-man published clinical trial last year. The future of presentation, though, will be the organ repair hospital, and the first one of its kind has recently opened its doors in Silver Spring. Those organ repair hospitals, they will not only facilitate to extend time by perfusing these organs, but they will allow to optimize a graft using bioengineering concepts such as gene editing, immune cloaking, or immune engineering in a controlled setting where the data is remotely accessible by the surgeon in real time, who assesses when the transplant is ready to be grafted into the recipient. So the future sequence of transplantation might change entirely from recovery and transplantation to recovery, optimization, and then transplantation. And this will, of course, open up the field for conditioning regimens for tolerance in cadaveric donors, innovative desensitization strategies, and will fundamentally change how we perform transplant from an acute setting towards elective surgery. However, not only advances in normothermic machine perfusion have been tremendous, but there have been also a great deal of advancements in the field of cryopreservation as well as in organ banking. And I know some of those concepts, they might sound futuristic or far-fetched, but I would like to share with you another video here from Ken Story, who impressively demonstrates to us that some of the most exciting technologies for organ preservation nature has already put in place, and it's upon us to decipher the underlying mechanisms to make the impossible possible.
These are the wood frogs that were caught last night.
The freezer is set at 8 degrees below freezing, roughly the temperature of the ground in winter. In a matter of hours, they'll be hard as rocks.
We have fully frozen Rana sylvatica wood frogs. They're probably 50–60% ice now. And their hearts are stopped, their brains are flatlined, their limbs can't be moved, they would be broken off. Yet they're still alive.
But the most amazing thing is the fact that the wood frog comes back to life. Over a couple of hours, it makes a miraculous and complete recovery.
So it is a fair question, why if the frog can do it, we still can't? But I think work that is currently performed by Kirk O'Duigan at Harvard with regards to developing new preservation solutions and advances in supercooling actually have brought us quite close to mimic some of those nature-inspired strategies when it comes to organ preservation. And similarly, there have been tremendous advances in the field of nano-warming that actually have shown that we are now able to prevent the formation of ice during the rewarming phase. And these advances in cryobiology have allowed us to cryopreserve and auto-transplant whole adult sheep ovaries and thereby preserve fertility. And this does not only have tremendous impact on restoring fertility for women, but also when scaled, could be a first step toward full organ banking. So the momentum and the opportunities here seem to be tremendous, and this is one of the reasons why an ever-growing number of clinicians, scientists, and organizations have built the Organ Preservation Alliance with the overall goal to transform research in this area in the future. Another area that will significantly shape the future of transplantation are gene editing technologies such as CRISPR-Cas9. And they might have striking potential to open new doors when it comes to immune modulation in xenotransplantation. George Church's group has recently shown that they were able to actually inactivate all the perfs in isolated pig cell lines, and eventually were able to create perf inactivated pigs using somatic cell nuclear transfer. And this is an important first step towards a safe applicability of xenotransplantation eventually in the future. Similarly, the technology of somatic cell nuclear transfer to generate genetically engineered organ niches has opened up the field for interspecies organogenesis. Using recipient-derived pluripotent stem cells and interspecies blastocyst complementation, we might be able to actually create human organs in animals that are closest with regards to size, anatomy, and physiology to humans. And these concepts have actually recently shown to be feasible in a large animal setting, where it was demonstrated that human pluripotent stem cells integrate and differentiate in pig embryos, although still at low levels and with low efficacy. But when optimized, this could be a first step to show that interspecies blastocyst complementation could be applied using human pluripotent stem cells. Of course, such strategies, they come with reasonable and yet unresolved ethical concerns when it comes to human-animal chimeric embryo research. But as this study shows, the vast majority of both the general public as well as the scientific community are very much in support of pursuing these strategies in the future. Engineering tissues or whole organs remains another grand challenge for the future of organ transplantation. Technologies such as 3D bioprinting or de- and recellularization of human-sized hearts and livers, or the use of full-scale bioreactors to grow organs from stem cells might be a viable opportunity in the not too distant future for our field. Impressive progress has been made in this area, such as bioprinting of 3D human renal proximal tubules, which represents a first important step towards the creation of complex renal tissue in vitro. Nanotechnology is another area that will revolutionize many areas in modern medicine, including transplantation when it comes to diagnostics and therapeutics. The number of nanoparticle therapies specifically tailored for immune modulation is vastly growing, which is fueled by not only advances in nanotechnology, but also a better understanding in the underlying immunological pathophysiology. And finally, the search of the liquid biopsy will remain an area of research in the future in our field. Advances in molecular diagnostics using the omics platforms, they have shown that we are able now to create unique biomarker profiles that can be correlated with rejection or underlying disease states. Molecular phenotyping or tissue-based molecular diagnostics, as pioneered by Dr. Halloran and the Alberta program, will significantly aid in improving patient care by improving diagnostic accuracy as well as a better understanding of the underlying mechanisms of these disease stages, much more so than conventional histologies. And as these platforms continue to mature and advance, I think we need to have a holistic approach, including big data concepts, to incorporate all the variable factors that might contribute to graft survival. So with this, I think that the technological capabilities and advances have never been larger before. We need to make sure that we utilize them to the best of our ability so that patients such as Helen, Seymour, Sonia, and Mark will also get a chance to benefit from the gift of transplant in the future. Like the pioneers in our field, we need to be bold as we embark and depart for the future of transplantation.
But if I were to say, my fellow citizens, that we shall send to the moon 240,000 miles away from the control station in Houston, a giant rocket more than 300 feet tall, the length of this football field, made of new metal alloys, some of which have not yet been invented, capable of standing heat and stresses several times more than have ever been experienced, fitted together with a precision better than the finest watch, carrying all the equipment needed for propulsion, guidance, control, communications, food, and survival, on an untried mission to an unknown celestial body, and then return it safely to Earth, re-entering the atmosphere at speeds of over 25,000 miles per hour, causing heat about half that on the temperature of the sun, almost as hot as it is here today, and do all this, and do it right, and do it first before this decade is out, then we must be bold.
Six, 5, 4, 3, 2, 1, 0. All engine running. Liftoff. We have a liftoff. 32 minutes past the hour. Liftoff on Apollo 11.
It's one small step for man, one giant leap for man.
So let's dare to be bold, and the future of transplantation might be a bright one. Thank you very much for your attention.
Gerald, Churchill said once, it's very difficult to make predictions, especially about the future.
That's right.
But you have done it remarkably well. We have one question which actually comes remarkably from a young, very promising trainee from your hometown, from Innsbruck. His name is Tony, and he sits someplace over there. And what he asks is, I adore transplantation, and I want to dedicate the best years of my life training to do it. What is then one area that if I dedicated my life's efforts to it, I will be really part of a big bang for the future?
I think it's one of the most exciting things that you can embark in the future. I think transplantation will be a culmination of the translation of all those exciting, promising technologies that we have seen, and probably be the era where we see a first in man to incorporate advances in tissue engineering, stem cell biology, or 3D bioprinting in the future. So I think this is one of the most exciting spheres you can go into at this point.
Thank you. Ladies and gentlemen, Gerald Brandacher.